which goes to Professor Edith Hurth. Professor Edith Hurth is a professor at the Collège de France, holding the chair of epigenetics and cellular memory, director of the Genetics and Developmental Biology Department, and group leader of mammalian developmental epigenetics at the Institut Curie in Paris. Like for George Church, it is impossible to do justice to all of Professor Hurth's important contributions to science in this brief summary. And like for Professor Church, I can refer you to Wikipedia for detailed information on her many achievements. But also, of course, you can read the interview with her in our program book. I will briefly mention a few things. She studied in Cambridge, where she did a PhD at the Imperial Cancer Research Fund in London, before she moved to France, where she pioneered the field of epigenetics and gene regulation. She is most widely known to many of you for her work on X-chromosome inactivation, and for doing this work she developed many innovative single-cell techniques for imaging DNA in cells, and is co-discoverer of the so-called topological associated domains, and studied their role in chromosome organization and dynamics. In 2013, Professor Hurth has given one of the opening lectures at our meeting in Paris, and we look forward to her talk again this year on X-chromosome structure and epigenetic dynamics causing X-inactivation. But first, I would like to present this year's ESHG award to Professor Edith Hurth. I will briefly read out what stands on the plaque for everybody. So the ESHG Award 2017 was awarded on May 30, 2017 to Professor Edith Hurth by the European Society of Human Genetics. The ESHG wishes to recognize Professor Edith Hurth for her groundbreaking contribution and pioneering work to the field of human genetics, epigenetics, and developmental biology. Her work, using innovative single-cell technologies, has shown how X-chromosome inactivation occurs as a dynamic process during early development. This has provided fundamental biological insight in the role of chromatin changes and chromosome organization in gene regulation. On occasion of the European Human Genetics Conference, Copenhagen, May 27-30, 2017. And now invite Professor Hurth for her talk.
Thank you very much. It's a huge honor to have received this award. And quite intimidating to be standing up here in front of so many people, and also to be speaking after George Church, who's one of my heroes. So I feel quite humble. So yes, I'm still going to talk about X-inactivation. I've been working on this process for almost 25 years or more, actually. And I hope you'll see that it's opened up many questions and continues to be a topic that I think attracts people from many different realms. So I think it's quite fitting that I should be speaking after the Mendel lecture, because in fact, X-inactivation can be considered as one of the many examples of exceptions to Mendel's laws. So in the case of genotype fitting with phenotype, there are several examples that were remarked over 100 years ago, almost 100 years ago, that actually are exceptional in the sense that the phenotypic variation found within the same individual is present, and this is due to gene expression differences. So I think you all know of Barbara McClintock's work on mobile elements, and actually some of her discoveries led to the idea that there could be controlling elements. This was, of course, DNA elements, but it turns out that they are indeed epigenetically regulated as well. Position effect variegation in Drosophila, and similarly, much of the work in fish and yeast has also shown that indeed there is variegation of gene expression and therefore cellular mosaicism. And X-Inactivation was one of the first mammalian examples of such exceptional patterns of gene expression within the same individual. And it was these sorts of examples that laid the foundations of what we term sometimes as the modern definition of epigenetics. So not so much the definition of Conrad Waddington, but more the definition of Riggs and Holliday, who in the 70s used this term to denote heritable changes in gene activity states that don't involve DNA sequence changes. And so the idea being that the cells within these individuals are genetically identical and yet are showing rather different gene expression states, and this is clonal and heritable. So I thought it was fitting that I should actually show you the paper by the person who really discovered X-Inactivation, Mary Lyon. This paper was published in Nature in 1961, and it was less than a page long. Those were the days. There were no figures. It was pure science, pure ideas and results. And she was really a remarkable scientist, a true geneticist, who realized that these variegated coat color patterns that she was seeing in mice and also in cats, it turns out, were related to females and to X-linked gene expression. And so she proposed in 61 that one of the two X chromosomes in females must be silenced early on in development, and that this silencing should then be propagated stably. I think, if I'm not mistaken, Mary Lyon was actually a recipient of this same award in 1993. So I'm truly honored to be receiving it myself today. So X-Inactivation, as I already mentioned, involves the silencing of one of the two X's. Now, there are different strategies in different organisms to deal with the sex chromosomes. I think in this slide, all I wanted to illustrate was that, in fact, males and females are actually rather similar. Some of the major differences are hidden under these leaves. There are other differences as well, in some other organs. But essentially, it's to do with the sex chromosomes. And what's essential for, in essence, to make a guy is on the Y. But the Y is a tiny little chromosome. It only has 100 or so genes, and it was recently shown that probably only a couple of those are actually truly essential for male-specific characteristics. But the X chromosome is much larger, over 1,000 genes, and having two copies of it versus one certainly does seem to be a problem. The imbalance in gene dosage is critical, and this leads to lethality. If dosage compensation is not achieved during early development, at least in the mouse, it's been demonstrated very clearly, this leads to very early embryonic lethality. And so it was Mary Lyon, therefore, that proposed, when she noted that one of the two X's is silenced, that this could be linked to dosage compensation. And this was also preceded by work and a hypothesis from Ono and others, but it was really Mary Lyon, I think, that clinched it. And so this is, again, just an illustration of what this is about. The idea is that initially the two X's are active during early development, and then one of them will become randomly chosen for silencing and will be shut down. And this state will be, as I said, clonally propagated, which is where the epigenetics comes in. And this leads to then clonal populations of cells expressing one or the other of the two X's. And having discovered this in mice and cats, she and others went on to propose and then to find that indeed in humans and other mammals, similar random X chromosome inactivation can be seen. And I just want to point to a paper from Jeremy Nathan's lab that came out a few years ago, where he made mice with fluorescent HPRT genes, a green version on one X and a red version on the other X, and then he looked at females. And these are three different pups from the same litter, and I hope you can see that they show very different ranges of green and red within them. So there is this mosaicism, but it varies enormously between individuals in terms of the spread and the degree. And this is actually a brain section, a coronal section of a mouse, one of these brains. And I hope you can see that the right hemisphere is completely green, so let's say paternal X is being expressed, and the red is, and the left version side is completely red, so the maternal X. So this was one example. There are many other cases where there's much more mixing, but just to show you that indeed the way the female brain is put together could be quite diverse in terms of parental X-linked gene expression. So here's a very simplistic view of what this process is about, and I'm going to plunge into some more detail soon. But it was discovered in 1990, in fact, that there's a unique molecule, an RNA molecule, that triggers this process of X-inactivation. So we already knew, decades of research had defined the critical region of the X chromosome that is necessary for this process. The X-inactivation center, shown here as these little red boxes. And EXIST was discovered by serendipity within this region and found to be only expressed from the X chromosome that would be silenced, and found to coat it. And this is an RNA fluorescence in situ hybridization, and you can see EXIST coating the X chromosome. And this coating very rapidly triggers gene silencing. And so here you can see an undifferentiated female ES cell where both X's are active. And in red, you can see the nascent transcription of an X-linked gene, so it's biallelic. And in green, you can see EXIST, which is expressed at very low levels. And as soon as differentiation is triggered, EXIST RNA will accumulate on just one of the two X's, will coat it, and will start to shut it down. So you start to see gene silencing very rapidly. And once this has happened, this state is progressively locked in thanks to epigenetic marks, chromatin marks. And in a very simplistic way, I want to show you what some of these events are. So this is a happily transcribing gene on the active X chromosome, and then EXIST comes along and switches it off. Now, for a long time, this was a bit of a black box. We now know, thanks to several papers published in the last couple of years, what some of the partners of the EXIST RNA molecule are, and they're quite surprising. And several of them are involved in RNA metabolism, and others are involved in chromatin changes. So these factors are brought in and very rapidly seem to change the chromatin and the gene activity of the X chromosome. And then it seems that the progressive recruitment of epigenetic machines, such as the polycomb group factors and other proteins, such as macroH2A, a histone variant, are brought in, and this is what progressively locks in the inactive state. And so we and others for many years have been studying, or still are studying, what the roles of these different factors are, how this proceeds. But one of the rather fascinating features of X-inactivation that came out over the last few years is the degree to which some genes can escape from this process altogether. And there are some genes that seem to be able to escape right the way through. In other words, even when EXIST coats, they still manage to be expressed from the EXIST-coated chromosome. So these we call constitutive escapees, and many of them are actually conserved between mammals. And it turns out that many of them are involved in chromatin-modifying functions. But maybe even more surprising was the discovery that there are some genes that can escape in what we call a facultative fashion. They get silenced first, it seems, and then become reactivated in a very tissue or lineage-specific fashion. And these genes are rather remarkable because indeed they go through this phase of being completely shut down and then somehow become very specifically reprogrammed or reactivated. And so this is something that, that many people in the field have started to become very interested in because it raises all sorts of questions about how genes are regulated in general, how heterochromatin is regulated, and also the extent to which gene dosage is important. And this is what I'm going to be telling you a bit more about today. And And so, yes, so as I mentioned, many of these SKPs, especially the constitutive SKPs, seem to be involved in chromatin, suggesting, and this is something that we actually proposed a few years ago, that some of them might even have to be expressed in a double dose because they have functions in X-inactivation. For example, the ATRX gene, it's a chromatin remodeler, and it's actually reactivated in certain lineages in extraembryonic cells, where in fact the ATRX protein itself seems to associate with the inactive X chromosome, suggesting that indeed you may specifically need a double dose of proteins, of some genes in some contexts. So, so this is just to tell you that in addition to being mosaics for paternal versus maternal X-linked gene expression, this escape phenomenon that was described some years ago and in a Nature paper by Laura Carroll and Hunt Willard, they actually described this in some detail. In different human individuals, the number of genes that escapes from X-inactivation can vary quite a lot. Up to 25% of X-linked genes can actually do this, and they vary, as I said, between individuals, and they can vary within tissues. In this paper, they only looked at lymphoblastoid lines, but as I said, we and others have looked and found that there is quite a lot more escape than we imagined. And so this quotation by Hunt Willard, I really like. As a man, I guess he's allowed to say these things, but genetically speaking, if you've met one man, you've met them all. Rather predictable, but women are far more diverse, and it's not just Mars or Venus, it's Mars or any other of these planets. So the take-home message here is everyone, of course, is unique, but girls are even more unique than anyone else. Okay, back to some more classical work, and I want to highlight the fact that although Mary Lyon discovered this process of X-inactivation, in fact, it had already been noted by Barr, a cytogeneticist, in 1949, that there was a heteropyknotic structure in female cat neurons, in this case, but in other female cell types as well. And it was also based on this, that it was proposed that indeed this might be the X chromosome, and it would be silent. And this is what electron microscopy from Andrew Belmont's lab showed is potentially the structure of the inactive X. So this is facultative heterochromatin because it's not there throughout life. It becomes reprogrammed in the germline. But nevertheless, once it's inactive, it looks pretty heterochromatic. But I also like this quotation by Cummings, who in 1968, in a rather premonitory fashion, suggested that indeed few structures in nature seem to be arranged with as much abandon as the chromatin of the interphase nucleus, and yet this random appearance may be deceiving because any degree of order would be well hidden by the very mass of the chromatin itself. And that's really at the heart of what we have been trying to understand for many years in my lab. What is this heterochromatic Barr body, and how does it relate to gene expression? And so I've told you that many genes seem to escape from X-inactivation. How do they do this? Where are they positioned, or how are they positioned on the X chromosome, and how is this escape driven? And so, in fact, over 10 years ago now, my lab and that of Jeannie Lawrence actually delved into the structure of the inactive X chromosome using the techniques that we had at hand at the time, which were fluorescence in situ hybridization and microscopy. And we looked at the organization of the inactive X relative to the active X. And this is a DNA fish where we're detecting in green several X-linked genes. In red are repeats, line 1 repeats. And in white, this is an RNA fish, so exist RNA fish. So this is the inactive X, and this is the active X. And I hope you can see that indeed the active X seems to be rather more dispersed, less assembled together, with the genes rather spread out, and the inactive X is rather well packaged. And this is work actually from the Kramer labs, where they even measured the overall compaction and volume of the inactive X chromosome. And it turns out it's not that much more compact than the active X. It's about 1 to 2-fold more compact, but it's not that condensed, which was rather a surprise. And what we believe is happening is that, in fact, what you see here is this dappy dense Barr body is mainly repeats. It's the repetitive fraction of the inactive X that really seems to be so highly condensed. And what we discovered in this paper 10 years ago was that as X chromosome inactivation happens, in fact, genes are located in a rather different organization during the process. So when Exist first coats the chromosome, as I mentioned to you earlier, transcription stops, but in fact, it stops first within the repetitive fraction. And so here you can see this is RNA pole 2, and where Exist has coated, you can see a sort of black hole where pole 2 is. Now, it turns out that the genes on the X have not yet been silenced early on, and they're located outside of this pole 2 hole, and they're still being transcribed. So this is an RNA fish here, and you can see an X-linked gene sticking out into the blue, which is pole 2. So the genes are still on as Exist coats, and you start to see this sort of black hole of transcription. And as X-inactivation proceeds, the genes become relocated into the repetitive core progressively. The only genes that don't do this are the genes that escape. Genes that escape X-inactivation always remain located on the outside or on the periphery of the inactive X chromosome. So this got us even more interested into the spatial organization of the X. And this is one of the reasons why we decided to start to use some of these technologies that I'm sure you've heard much about at this meeting, chromosome conformation capture. And so I won't go into too much detail because I know that this has been introduced before, but there are many different variants of this technique, which was pioneered by Job Dekker, and basically what it entails is to cross-link DNA within a nucleus to capture any of its nearest neighbors, to then cut with a restriction enzyme that will allow one to capture after ligation, and then either sequence whole genomes, although that actually can be quite expensive, or to go in and zoom in on specific regions. And so it was thanks to these technologies that many of the different scales of organization of the genome have been discovered. And I want to just say a couple of words about this discovery of topologically associated domains, which we were one of the labs together with Job Dekker's lab that we were lucky enough to stumble on this organization, although it confused us for many years. So we were actually on a hunt for the X-Inactivation Center and Exist's regulatory landscape. And I won't go into details about this. It was published and we're still carrying on trying to dissect out what all of this meant, but basically what we did was a chromosome conformation capture 5C technique across an almost 5 megabase region, and this was Elfej Nora in my lab who actually who set this up. And when we did this 5C approach, what we discovered was that this region is organized into these large domains of interaction that we termed TADs because we thought it was an easy term to remember. Maybe we shouldn't have called them TADs because maybe some other kind of domain will end up becoming more interesting or important. But in any case, topologically associating domains are essentially regions of the genome that have a higher frequency of meeting together locally than regions beyond them. And so you can see this mapped out here in one of these typical heat maps where basically you have these large domains of interaction. And the region that interested us most was this X-Inactivation Center region, which we discovered was very unusually organized. Exist and its 5' region was organized into a 500 kb TAD, within which all of the known positive regulators of Exist are located. And the antisense repressor of Exist was organized into this smaller TAD. And basically, we validated this by FISH, high resolution or super resolution microscopy in FISH, and we were able to show that indeed Exist communicates more frequently with its 5' positive regulators than with its 3' antisense repressive TAD. And so this opened up many interesting questions, and I think some of them have been addressed during this meeting. But one of the first layers that it opened up was how this level of the genome is organized. So I'm sure you've seen this cartoon many times. We have the genome, and now we're still trying to figure out how it's folded. And this is where we think TADs filled a gap in terms of the organization at the sub-megabase scale. And it's at this scale where gene regulation happens. And so the regulatory landscapes of genes tend to lie within individual TADs. And genes that lie within the same TAD can often be found to be coordinately regulated in developmental contexts, for example. And so if you're hunting for your favorite enhancer, for example, you're most likely to find it within the TAD that hosts the gene. So this was all very exciting, and it distracted us for quite a time, I have to admit. We were very excited to have seen this kind of organization and try to understand how it relates to how exist is regulated. Bing Ren's lab also discovered this at the genome-wide scale and showed furthermore that this organization is conserved, or at least the boundaries between TADs seem to be conserved between mouse and man, suggesting that it probably is an important level of organization. But we got distracted from our central question, which was, well, what about the X chromosome when it's inactive? And we did a poor man's experiment at the time, because 5C, I didn't mention this, but with 5C, you cannot get allele-specific information. You sequence the oligos you use to do the C technique in this particular example. And so we did 5C on male and female ES cells and on fibroblasts, and this is what I'm showing you here. So these are the female fibroblasts, the same region, male fibroblasts, and of course in a male you only have an active X, and the female you have the active and the inactive X. So as I said, we did the poor man's experiment, we just subtracted one from the other. The reviewers hated it, of course, but we didn't really have any choice, because at that point, high C wasn't easy to do, or at least not at this resolution. And so what we found was that indeed it seemed like the map after subtraction was rather flat, that there wasn't that much structure on the presumably inactive portion of the X. But we decided to do this properly then in collaboration with Job Dekker. And what we did was we exploited F1 hybrid ES cell lines where we knew what the parental origin of the two genomes was, and they're highly divergent in terms of sequence polymorphism. So 129 Castaneda's cross, which gives about one SNP every 100 or so base pairs. In fact, the X chromosome, it's less than that, sadly, but still, it gave us quite a good way of getting allele-specific information. Now, we differentiate these ES cells, and of course, X-Inactivation is usually random, so you end up with this population of a mosaic cell population where either one or the other of the X's has become inactivated. So in order to get allele-specific information at the molecular level, we had to subclone. So we then subclone cells, and in this case we decided to differentiate into neural progenitor cells because they're quite easy to subclone subsequently. And we isolated multiple different clones. And then we performed RNA-seq, high-C-seq, and attack-seq. And this is just to show you the RNA-seq on 4 independent clones, 3 of them, and so this is showing you expression from the 129 chromosome. So when you're only expressing the 129X, you're red. When you're only expressing the castaneous allele, you're green. And if you're biallelic, you're black. And you can see that in these 4 clones, you have only the castaneous X inactivated here, and here you have the 129X chromosome inactivated. But I hope you can see that there are some regions that are biallelic. And these are the regions that show escape. So we decided to look at one or some of these clones more carefully using high C. And I should point out that when we zoom in on these regions that escape, we realize that they're often in clusters. So we have these constitutive escapees, but we also have clusters of facultative escapees. And I'll come to that in a minute. So we performed high C, as I said, on these NPCs, and this is what we found. And actually, this paper was published last year, and I realized as I went through my talk, and it was too late, the reference isn't in there, but it's Giorgetti et al., 2016. And so basically, we performed the high C in these NPCs and then looked at the active X and the inactive X using the allelic information. So this is the whole X chromosome here, and I hope you can see that indeed there's organization into compartments. If you zoom in a bit, you can start to see the TADs appearing very nicely here. here. These are the TADs that I mentioned earlier. This is 40 kb resolution. But then, if you look at the inactive X, the first thing that struck us was that you have this organization into these two huge domains of interaction, and you don't seem to have the compartmentalization that you see for the active X and for the rest of the genome. So two, what we call megadomains, they've also been called superdomains, and separated by this rather unusual region, DXZ4, that I'll come back to in a minute. And so this organization at the chromosome-wide level was also paralleled by an unusual organization more locally. What we found was that the X chromosome, when it's inactive, is globally TADless. TADs seem to be lost more or less across the whole X, except at a few regions, and I'm showing you one such region here, which do seem to form TAD-like structures. So this is just to summarize what I just told you. Indeed, the inactive X is divided into these megadomains. We could also find that constitutive escapees that we know always escape actually interact at long range in trans, and I don't know if you can see these little spots of interaction up here. These are actually some of the constitutive escapees that somehow come together in nuclear space, but the rest of the X seems to be rather randomly organized, not meeting together in TADs, except at a few regions, as I mentioned. And it turns out that these regions are regions that escape. So here I've just flipped over the map so that you can, and I've zoomed into one such region, and this is a region of a cluster of several genes, within which lies a gene that I'm sure many of you are very familiar with, MECP2, which of course is implicated in Rett syndrome. It's a methyl binding domain protein, and it's a gene that classically is thought to be completely shut down and actually very difficult to reactivate, even in tissue culture. And here we found that MECP2 is biallelically expressed. It's expressed from the inactive X. So this is expression down here. This is a TAC-seq here, and this is the inactive X. Up here you have the active X. So you see biallelic expression of MECP2 and several of its neighbors. And when we look at the TAD organization, what we realize is this TAD-like structure on the inactive X corresponds very closely to this cluster of escaping genes. And when we look at some of the other regions on the X, exist, for example, is only expressed from the inactive X, and it doesn't seem to form a TAD-like structure. This gene alone doesn't seem to be able to create any kind of structure or to be present in a structure. So not all escapees seem to be in these TADs, but some of them do. Now this was also a bit of a surprise to us because indeed TADs are thought to be rather constant through differentiation and in somatic cells, but it seems that on the inactive X at least, they do seem to disappear globally, but then reappear in some situations. So we also looked at what regions seem to be accessible at the chromatin level, and what we found was that there seems to be a significant enrichment of TAC-seq accessibility at CTCF sites close to the SKP genes and at the boundaries of these SKP clusters. So this suggests that indeed there is some expression from the inactive X. It does seem to be organized, and maybe CTCF could have some role here. And this led us to propose some potential models. So one idea is that the active X chromosome that I'm showing you here is a series of blobs which are supposed to be the TADs and are being expressed, which is shown in green. As X-Inactivation happens, most of the X loses expression and TAD organization, but a few regions manage to maintain structure and expression. On the other hand, it could be that you lose expression, but you keep TAD structures, and then escape is facilitated by the presence of these TADs later on. Alternatively, you could lose structure overall on the X, and then escape happens in some transcription factor or driven fashion, and this is then what causes TADs to reappear. So we still haven't completely cracked that, but I want to now show you a little bit about what we've managed to learn when we looked at the dynamics of these genes. So using ES cells that we've differentiated first into epiblast-like cells and then into neural stem cells, we find that, for example, MECP2 seems to be shut down completely, as we had expected, during the first days of differentiation, but then later on becomes re-expressed, becomes biallelically expressed in neural stem cells. So we feel that we can rule out at least this part of the this model, but we can't distinguish between these two. So now I'm going to tell you a bit about the megadomains, and you'll see that this is related then to maybe to what facultative escape is about. So this organization into megadomains we found very intriguing, and we decided to explore what could be behind it. So first of all, I'm just going to summarize what we did to validate that these megadomains actually did exist in single nuclei using DNA-FISH, which is our favorite validation approach. So we used 18 megabase oligoprobes that spanned either that lie within one of the megadomains or that span the boundary between the two megadomains. And what we found, if you take the A and the B probes, is that on the active X, they don't seem to show any difference, but on the inactive X, they seem to be much more clustered than on the active X. So basically, we think that indeed there is more co-localization of sequences on the inactive X, suggesting that there truly is this kind of segregation into these two large domains of interaction on the inactive X compared to the active X. So, so this was something that actually validated the high-C data very nicely. So this is the high-C data, this is the FISH data. But we were then interested to find out how these domains actually form. And so to do this, we first of all explored whether EXIST could be involved, X-Inactivation itself. And so we used an embryonic stem cell system where we can induce EXIST expression even in undifferentiated cells. Then we can assess using FISH and also using high-C the degree to which structure changes. And what we found is that when EXIST is wild type and able to coat and silence the chromosome normally, you induce megadomain formation. And this is again using this FISH assay to show that there's more interaction with the A and B probes than with the B and C probes. However, when we used a mutant form of EXIST, that can no longer silence the X chromosome, and I'm not showing you this here, but it's in the paper, we found that this no longer happens. So you need EXIST-mediated silencing in order to trigger this megadomain formation. We also tested whether the boundary within or between the two megadomains might be important. And so now with CRISPR-Cas9, this was a trivial experiment to do. We deleted the 200 kb region that seemed to cover this boundary. And within it, what we noted was that there is a very unusual region known as the DXZ4 microsatellite, which is a series of repeats which is conserved on the human and on the mouse X. And I forgot to mention, but these megadomains also exist on the human X, and it was Lieberman-Eiden's lab who actually first described this for the human X. And this DXZ4 region seems to be essential for this organization in both cases. So it's a very unusual region. It's very enriched in CTCF, which is this rather magical protein that does seem to be involved in creating architecture on chromosomes. And it's actually euchromatic on the inactive X, and it's heterochromatic on the active X. So very intriguing. So when we knocked it out, we did the same experiment that I showed you earlier. We made knockouts in ES cells, then we differentiated them into MPCs, and then we subcloned and did high-C and RNA-seq, etc. And what we found was, so this is the active X. This is, I'm just showing you in parallel here, the wild-type version versus the DXZ4 deleted version. There's no difference in the organization of the active X when you delete this region, but I hope you can see that on the inactive X, whereas in the wild-type situation, you can very clearly see these megadomains, on the, in the deleted version, you no longer have this organization at all. So we were very excited. We thought, well, this, this is the region, and it really does matter for the formation of these megadomains. And so, of course, we looked to see whether there was any dramatic phenotype. And then we got slightly less excited because basically we saw that indeed the X was pretty much inactivated. It seemed to initiate inactivation normally during differentiation. We didn't see any change in the overall compaction of the X, and I'm not showing you this, but that was done by DNA-FISH. So we were a bit disappointed, but then we thought, well, maybe it still is having some effect on X-linked gene expression, and so we looked more carefully. So we went in and looked at several genes that are either silenced or escape from X-inactivation. And here, I'm just showing you the RNA-FISH data. We also did a lot of pyrosequencing and RNA-seq data. And basically what we found was that genes that show facultative escape are less able to escape in the DXZ4 mutant situation. And so here, I'm just showing you the percentage of signals that are biallelic in green or monoallelic in purple. And in a wild-type clone, you see there's quite a lot of escape for several of these regions, these facultative escapees. So these are 3 different regions here, but then in the knockout version, you see that this really seems to be dramatically less, so less escape. Now, constitutive escapees don't seem to care. With or without DXZ4, they seem to be able to escape. So there does seem to be an influence then of this region on facultative escape. But it wasn't dramatic. We did this many, many times in many independent clones, and what we found was that this facultative escape can vary quite a bit from one clone to another, a bit like what I told you earlier about human individuals and these lymphoblastoid cells. So there seems to be a variation in the degree of facultative escape, even in a wild-type situation, but there did seem to be a tendency to see less escape in the DXZ4 mutant. And so this actually allowed us then to explore the question of whether or not facultative escape is linked to the presence of TADs on the inactive X. And so here, I'm showing you the wild-type situation again. This is for the MECP2 cluster, where you can see on the inactive X there's this small TAD. But then, when we look in the DXZ4 mutant inactive X, where escape is no longer seen, you see that this TAD has completely disappeared. So this suggests that indeed, the capacity to form TADs and the capacity to escape are very, very tightly linked. And perhaps that TAD formation could be facilitating escape, or that whatever is driving escape is helping create TAD formation. So with respect to DXZ4, though, this is what we're speculating. So this region of the chromosome, as I said, is conserved and highly enriched in CTCF4, CTCF. And so what we're thinking is that during differentiation, maybe this region is interacting transiently with some of these potential escapees and helping them to escape, facilitating their escape, maybe by providing them with CTCF or something like that. So this is now what we're testing. But indeed, what we think is that DXZ4 is having some long-range influence on the capacity of some of these genes to actually get away with being expressed. And we also noted that these regions that show this facultative escape are also regions that never seem to be associated with the lamina. So we think that this could also be something to do with their freedom then to maybe interact with DXZ4. So just to conclude, and then I'm going to give you one last small set of data that carries on from this. I'm almost done. So basically, I hope I've convinced you that the inactive X is fascinating, not only in its expression, but also in its structure. This unusual organization where TADs seem to be gone, and it's organized into these 2 megadomains. How DXZ4 is doing this segregation or partitioning of the X is still, I think, an open question. We know it's important, but is it because it somehow tethers the X to some nuclear structure, or else changes its overall organization in some way? And these TAD-like structures that we see on the inactive X in NPCs, as I said, correspond to these facultative escapees that are clustered in many cases. And here we also think that CTCF might be involved in allowing these genes to escape, and maybe it's DXZ4 that is somehow influencing them. So, but I also want to point out that this was extremely variable, and we were plagued by the doubt that maybe this was just some kind of tissue culture artifact. And because half of my lab does in vivo work, we decided to attack the in vivo side of this very rapidly. And this now is what we're going to do now. So there are many questions open. I'm going to skip those and just show you some of the latest data that we've been able to produce, exploring how DXZ4 and how escape might be related. And so here I'm just showing you one of the tissues in which it's easy to explore escape in vivo. This is in extraharmonic tissues, in the trophoblast giant cells, where we were able to show that up to 20% of X-linked genes show some degree of escape. And it does vary, and so this is during development at around day 7. You can see that a lot of genes show expression from the inactive X chromosome, and so this is shown here by RNA fish. And so what's nice about TGCs is that during early embryogenesis in the mice, it's always the paternal X that gets silenced. I didn't want to confuse you with this information earlier, but basically X-inactivation is initially imprinted, at least in X-remnant tissues. It later becomes random. But in this imprinted form, we know that the paternal X is the one that should be silent. So we generated knockout mice for DXZ4 and crossed them to wild-type females, and therefore were able to address to the extent to which we could see escape in the DXZ4 mutants. So we've done this by RNA-FISH, and we're currently also doing it by RNA-seq, using polymorphic mice. And basically what we find is that even more than in the MPCs, we seem to see that the absence of DXZ4 does lead to a reduction in escape. So here in blue is the percentage of cells showing escape in a wild-type situation, and in red is in the DXZ4 mutant context. And I hope you can see that for these 3 facultative escapees, we definitely see reduced escape, whereas for a constitutive escapee, again, it doesn't seem to care. It shows escape quite fine. So this is in TGCs, but of course, as one of our favorite genes was MECP2, I'm sure many of you have been asking yourselves, well, how does that work? If MECP2 escapes, why is it involved in Rett syndrome, where only girls survive, and with a mutation, even the 50% or so of cells that are expressing the wrong X chromosome lead to this very, very severe disease? So we looked in vivo to see whether we see MECP2 escape as well. And so in early postnatal mouse brains, we could find biallelic expression of MECP2 in about 20% of cells, 20 to 30% of cells. And all these cells were SOX2 positive cells. So in the subventricular zone at around at postnatal day 6, we could see significant MECP2 escape. We then looked in adults, or that's what we're doing now, and here we also see some escape. It seems to be to a lesser extent and in different cells. And it's actually very difficult to perform this analysis by RNA-FISH. So the most recent set of experiments that we've been able to produce, in collaboration with Adrian Bird, who generated MECP2, GFP, MECP2, cherry mice that we've been able to use, we've been able to go in and look at both sections and fact sort different parts of the brain to assess the degree to which we see biallelically expressed MECP2 protein. And I'm not going to go through all of the details here, but suffice it to say that in several regions of the brain, we do see a percentage of cells that show biallelic expression. This is a brain section showing you that overall most cells are either red or green, but the occasional cell definitely is yellow. And using special flow cytometry techniques, we can now purify these cells and actually explore what exactly is going on inside them. So this is what we're doing right now, and of course, I'm sure what you're all asking yourselves, and I wish I could give you the answer, but I can't today, hopefully soon, is what is the influence then in the DXZ4 mice? To what extent do we see lesser or more escape? And what could be the phenotypic consequences, if any, of this kind of escape? So this is ongoing. And I hope that with this I've convinced you that the inactive X is definitely not as silent as we imagined it to be, that the potential to reactivate genes in some contexts naturally means that there could be a potential there to go in pharmacologically, and that maybe this opens up the possibility that one could help or stimulate the escape of genes such as MECP2 in a rather targeted way, particularly to help in syndromes such as Rett's, but obviously there are many other X-linked genes that are involved in other syndromes where this could potentially also be of use and importance. So with that, I will end, and I really want to thank the people in the lab who set this up. Alfej Nora, who has now left the lab and is doing a postdoc in San Francisco, but he was really the person who was behind the 5C that I showed you, together with Job Dekker, who's a fabulous collaborator. We've been collaborating for many, many years, and it's wonderful to have someone like him joining forces for the X. Luca Giorgetti, who is the postdoc who performed much of the work on the inactive X analysis, and he's now a group leader at the FMI in Basel. And then several other people, Lucille Marion-Paul, who's a neurobiologist and has been doing all the MECP2 work, and Michael Attia, who was one of the postdocs who did all of the genetic manipulation of DXZ4, and Catherine Corbel and Michael, who did the work on the TGCs. And Adrian Bird, of course, for providing the MECP2 mice, and Howard Chang for his help with the ATAC-seq. And with that, I'll thank you, and maybe have one question. Thank you very much. Thank you.
Thank you so much, Professor Hurth, for this fascinating presentation. There is time for one question. Is there anybody asking a question? Yes.
Hi, thank you very much for this amazing talk. I'm Bianca Tesi from Stockholm, and I would like to ask you what is known about the variability of this DX-zeta4 region in the human genome, and whether like a size effect or the specific of these repeats might explain why the number of facultative genes that explain that escape X-inactivation is variable among individuals.
Exactly. You hit the nail on the head. That's exactly what we're thinking and hoping. So in humans in particular, the copy number is quite high. It's about 100 copies, and it's extremely variable in the population. So indeed, it could be that the number of copies of DXZ4 will have a direct influence on the degree to which we see facultative escape. Now, actually, Erez Lieberman-Eiden also deleted this region, but he did it in somatic cells. And when you delete DXZ4 on the human inactive X in somatic cells, he apparently didn't see any effect on escape. But as far as we're concerned, it's too late, because we think that this region is working in a transient way during development. So, well, we're not doing CRISPR knockouts on humans, that's for sure, but we are hoping to explore this in culture, now that we know that, at least in the mouse, what we saw in tissue culture, we also see in vivo. So yes, that's a very interesting and exciting question, but I don't have the answer.
Oh, thank you.
Okay. Thank you.